quickly introduce myself. My name is Malcom, and I've actually quite recently joined Regulation to work in part with AQUA-FAANG and in part with, I think, a sort of similar consortium called Gene Switch, basically looking at not so much on kind of the annotation itself, but kind of new methods for improving stuff. And I'll get to that a bit at the end when we kind of ask what we actually do with all these peaks and all this data. But for now, yeah. So I guess we've got everyone here. Yeah. So just go ahead and introduce this tutorial. Really, the idea is that obviously, like when you've got peaks like you generated with Ilias earlier, it's not immediately obvious what to do with them, really. So this tutorial is going to take you through Diffbind, which is a program basically used to analyze whether something's more or less bound. I guess by a transcription factor, it would kind of be bound. By histone mark, it's more like a differentially acetylated or methylated. But basically, if there's any kind of real kind of quantifiable enrichment. I've noted here that kind of peak callers are kind of good for finding kind of regions of interest, but it's not the most like rigorous way to analyze something. So often if you've generated a set of peaks, if you're kind of really worried about whether the peaks are different between cell types or enhanced off in one cell type and on in another, using something like Diffbind is a really good way to make sure that you're not kind of, it hasn't been an artifact of the peak caller. It's not some kind of strange thing that's happened. It's actually something that's robust. And it should be worth noting that you can do this kind of analysis with other forms of annotation, right? You don't have to be using peaks from enhancers. You could use promoters, for example, or any other kind of annotation you're interested in, or any kind of transcription factor chip peaks or anything like that. I think in the original paper they have for Diffbind, they used transcription factor binding peaks at promoters in certain kind of cancer samples to look at differential drug resistance. I think that was the idea, basically. So yeah, and I've put a sort of like a big asterisk to the next thing. So we'll cover this a bit at the end, but basically, you can use this kind of analysis to link peaks or differentially bound peaks in nearby genes, but there's a ton of caveats that come with that, and it's quite it's not a trivial thing to do. So we'll come back to that later. Just to go over the overall view of the data in the tutorial, we'll go over the actual programming stuff a bit later. But we're basically looking at panels B and C from this figure from the paper, the same paper that the data with Ilias was using in the previous tutorial. And we're going to look at basically recreating this panel B, in not the heat map itself, but this kind of classification of peaks into sperm-specific and oocyte-specific. We're going to do that kind of more rigorously with Diffbind, and then we're going to look at how you might recreate a box plot of sort of FPKM levels at the enhancers as well. And then we're going to look at some of further analyses that can work with that. Yeah, and if anyone's, I haven't included anything about these kind of hit maps here, but if anyone's interested in these, I can link to some code that will generate plots like this, if that's something people want to do either now or later. I should say I'd be very welcome to people to ask questions either in the chat or just by interrupting with the mic, because there's not actually a huge amount of content in the this tutorial is probably about half and half kind of code and sort of just discussing ideas around kind of chip-seq analysis. So if people have questions, it's fine to ask them in the middle of the talk. I don't think we'll be tight for time with an hour and a half, fingers crossed. So yeah, let me know. Okay, so yeah, so to do this kind of differential binding analysis, it seems quite straightforward, but there's quite a few things you have to do it right. So fortunately, the way Diffbind is written, most of this stuff will just run automatically. You don't actually need to manually do all these steps. We've got that in the tutorial just so people know what's going on, because I think it's important to build up the intuition of what's going on behind the scenes. But in the background, yeah, there's all this stuff runs in the background. Diffbind will actually do these things in one single command if you want it to, if you want to be super lazy. And that's all in the Diffbind sort of materials and tutorials and stuff. But very simply, in order to sort of compare peaks rigorously between two cell types, First of all, obviously, you need to read in all that data and annotated data for samples and tissues. But you need to then collapse all of those different sets of peaks down into a single consistent consensus set of peaks across samples. We'll look at how that's done precisely later. You might want to exclude regions that look dodgy. Then you're going to need to go back to every individual sample, and now with the consensus reads, check how many reads, with the consensus peaks, check how many reads are in each of those peaks in each sample. And then you're going to need to somehow normalize these counts and then test the significance between samples. So the last two steps will be familiar to anyone who's done any kind of RNA-seq analysis. I hope that's a few people here. I'm not entirely sure exactly what we've got. I haven't been able to chat to too many people. But actually, Diffbind simply uses DESeq2 or EdgeR to do this step, because we'll cover this a little bit later, but there's obviously some quite kind of there's some real heavy lifting involved there to how you normalize this kind of short read count data between different samples, so you don't end up with too much noise. But that's all handled for you by the package, so you won't notice that you're using these programs. But probably if you're familiar with them, you'll see some similarities at certain steps. So just to say, is everyone kind of with me so far on what these ideas are? What's kind of the point of this whole thing? Does this kind of background make sense from what you've learned today and yesterday? If there's any questions.
When we use these GenRich for peak calling, that's already done as we had done for several samples together. So there's already some kind of a building and consensus between samples then. You go with this peak calling then from the already, in a way, consensus peak calling into this Diffbind?
So what I think you've done with Ilias is you've called peaks using several replicates of one tissue, right? Yeah, so I mean that makes perfect sense, and that's a really good way to generate robust peaks. But we're actually going to unify those peaks between sperm and oocyte in this step, because it doesn't make sense to call peaks using this kind of replicate-based calling when they're different samples, because you'd expect them to be different. Does that make sense?
Yeah, I mean, you could also create some kind of a overlap collection of peaks from the individuals, from the individual samples. So in a way, you're having two levels of unifying the peaks.
Yeah, I think that's correct. Yeah. There's actually Diffbind does allow you to do that. In fact, I'll come back to that in a second when we look at the peak calling sets. But yeah, it's entirely correct. Cool. So the first step, this is before generating the peaks, it's just to read in the files basically to Diffbind, and it'll be clear when you see the code what's happening here, I hope. But it's worth noting that Diffbind uses what it calls a sample sheet, which is just a kind of a comma separated values table, really, like a flat file table, something that could be an Excel document, if that's an easier way to think about it, a spreadsheet, which just has one line per input. So it has the BAM file with the counts, the sample name, any kind of data like tissue, replicate, anything like that, and the peak calls to use for that sample. But that's handed in as a table rather than as an argument to the program, which is an interesting way to do it. It certainly makes sense. If you have lots of samples, you wouldn't want to be doing anything else. And that's actually quite a good way. If you find yourself writing kind of reusable programs or pipelines, this can be quite a good way to do it rather than passing lots of command line arguments. I think you guys all know what bed files are, hopefully now, from the tutorial with Ilias. And yeah, so this is exactly like your question, actually. Sorry, I didn't catch your name, but it didn't come up on Zoom. But it's exactly like the question that was asked. We have them for each tissue rather than each sample individually. So we've already got one level of robustness there. We also still need the BAM files. We don't just go from the annotation of the peaks for this. We actually do need to find out exactly how many reads a bound there. As we'll see, the regions of the peaks might change slightly as well. I don't know how familiar people are with BAM files. They can be a little bit tricky because they're compressed files, so you can't read them directly. And they're also indexed, which means in theory, you can read parts of the BAM file without reading every single thing in the BAM file, which is really useful for this kind of analysis where you've got peaks, so you just want to go in and take a tiny bit of the file. So the ones in this tutorial are actually downsampled, so they're kind of a tenth of the coverage of the real ones. But the original files provided with the paper, or rather the original reads, generated sort of 15 gigabyte BAM files, which is really quite big if you want to read it all into memory. And we also have some simple metadata included in the sample sheet. So for us, it's just tissue and replicate. Tissue being either sperm or oocyte, and replicate being just one or two. So now we have to generate the peak set from these consensus peaks, and there's basically 3 steps to this. We merge the peaks, and this function, as you'll see in the tutorial, has an argument called minimum overlaps. So assuming we called each sample separately, so we had 2 sets of peaks for oocyte and 2 sets of peaks for sperm, we might want to do something where we say, well, actually, we need to have at least 2 peaks on a region to count it as a peak. Sorry, at least 2 peaks together to take that peak into the consensus peak. Because if you've seen it in one replicate and not the other, you might assume it's a false peak. That's been handled for us by Genrich, basically, in this tutorial. So we're not going to require that. Obviously, it would make no sense to require a peak to be present in both sperm and oocyte for further analysis, because that would kind of ruin the point of the comparison we're trying to make. So as you can see, that this process is actually quite crude, the merging of peaks. It really just takes the overlaps of each peak. I hope this diagram makes sense, and turns it into a big peak. Then I'm not entirely sure how this works internally, but roughly speaking, what Diffbind does is it calculates the summits of the peaks, which I assume is based on the highest level of kind of pile-up signal. I hope it doesn't do something silly like just kind of take the center or whatever. And then it recenters this peak to being 200 base pairs around this peak. And obviously, having a consistent length of peak is important if we're going to make comparisons, or rather, it would be an extra thing we'd have to normalize for if we didn't sort of fix all the lengths of the peaks to be a set value. You can change this 200 base pair mark. That's probably reasonable for the marks we're looking at today. That would be sort of basically 1 or 2 nucleosomes worth of enrichment, but that might change depending on some transcription factors would actually be much narrower than that, and some marks can be very, very broad, and you might want a larger peaks. It's just something to consider. So one step that's not in our tutorial today is the use of a blacklist for ChIP-seq. Now, this is a bit funny because you might think that because we have all these replicates and if we have control ChIP-seq and this kind of things, we wouldn't need to do this. But there's just certain regions of the genome that are very badly behaved with ChIP-seq. They tend to be sort of subtelomeric or like centromeric regions. You can see from the graph on the right that I stole from someone on Bioconductor Support Forum that you just have these huge peaks in enrichment at the telomeres and at the centromeres. I'm not sure anyone really knows why this is. It's probably to do with just reads. Oh, sorry. Oh, goodness. This laptop has a touch screen and I got some dust on it and that's ruined everything. Yeah, you can see there's a huge amount of enrichment at the centromeres and the telomeres. That's the middle and the end of the chromosome. So yeah, it's possible to generate what's called a blacklist or a grey list using the control ChIP-seq data. And I haven't covered how to do that in this tutorial, and I'm not sure whether you guys will be generating. That's the point, actually. Does anyone know if AQUA-FAANG are generating controlled ChIP-seq tracks? Okay, maybe not. But if you guys are,
with control ChIP-seq, you mean just the input without?
Yeah.
We will have inputs for our sample.
Okay, cool. Well, it's good to have a kind of generate a blacklist from that if you can. Maybe I'm just talking to I guess we'll be doing that, maybe, I don't know. So, but that's an important thing, just because otherwise, I've seen this happen in lab meetings during my PhD. Someone would get a list of kind of top enriched peaks, and they would all be just like right at the end of the chromosome and just obviously all wrong. They'd be 5 kilobases long and nowhere near any genes or anything. So yeah, it's an easy way of doing that. And there's a paper. Yeah,
I was just going to ask, do you have any recommendations for any softwares or packages to create these blacklist files?
I don't. I've just put a reference here to a paper, which now I think about it, I haven't actually uploaded to the Google Doc, but I will.
All right,
it's open access, so yeah.
Thank you.
Another question: if you run already GenRich with the can you run GenRich already with the control file? Then this should also consider this problem, or isn't it?
Yeah, it seems like often these regions are enriched over control, and I don't really understand why, to be honest. But that is what you're seeing on the right is actually a graph of enrichment over control. I don't know why it is that this still persists, but it seems to be the case. Yeah, funnily enough, it actually doesn't solve that problem. Yeah. There might be some discussion in the paper. I'm not sure. Sorry. Yeah. Another question.
If you don't have any control, then what? Then these peaks are not generated, or what? How to avoid this thing?
So if you don't have a control, so blacklists are not particularly experiment specific, right? So if you don't have a control, you could probably just go and download one. You could certainly do that in sort of human or mouse, probably in zebrafish as well. I haven't done that for this tutorial because I didn't find one in time, but it's probably out there. And if you don't have a control, to be honest, a lot of these things will just mess up your analysis. Like you'd find peaks in strange places that look odd. I think you probably wouldn't. It wouldn't completely undermine what you're doing. It just would create a bit of noise, a bit of extra noise. Does that answer your question?
Yeah, but so without control, then it doesn't make any sense to do this kind of experiments, or what?
No. It's still, the experiments still work without control. It's just better to have one. Yeah.
Okay, so we have a larger peaks at these regions if you don't have any control.
Yeah, you could end up with peaks in repeat regions if you don't have a blacklist. Yeah.
Okay.
Yeah. It's not something to worry about hugely. I think, especially if you've looked at a bit of this data, it's often quite obvious when this is happening because the peaks look very strange and they're very highly enriched, and it's kind of just everything looks a bit wrong if you open it in a genome browser like you did in the last tutorial. But it's just worth noting that this can help your analysis. Basically, it can help it go a bit smoother.
Okay. Thank you.
Cool.
Sorry,
so then, yeah, go ahead.
So I just missed or didn't understood correctly the answer to give to Krista, because I never used well, it's the first time that I'm using GenRich. I've used Max, and when you're already taking into account the inputs sample for the peak calling, it kind of solves the problem. So you're proposing that we would deal with the inputs sample after the peak calling, or during the peak calling and after? I didn't understand that. No.
So you would call the peaks using the input, sorry, using the control and the signal track as you've done. That makes perfect sense. This is an additional step that ENCODE found that they had to go through to filter out certain erroneous peaks. So even using the control, they were still getting peaks called against these really strange regions at the end of chromosomes. And that's why they generate the backlists. So it's an additional step. Even if you're using a control,
okay,
this is still a good idea.
So we do it twice. Kind of.
Yeah, basically. So you actually, the way it would work is that you generate the blacklist. I believe, sorry, I'm not super hot on this. I believe that you just use the control track to make the blacklist. Yeah. And so that would just be a kind of a filter step. Yeah.
Okay, thanks.
Cool. Hopefully that reference is useful. It's quite a recent one. Yeah. Cool. So now it gets a bit simpler, really. Now we have our consensus peak set, and we've maybe excluded some peaks for the blacklist. We just have to count the reads, and this is the step that uses the original BAM files. And yeah, very simply, it's the dba.count function which does this, and this will also generate the peak consensus peaks for you as well. It's quite a Diffbind just kind of holds your hand quite well, I think, through these analyses. And it basically reads in per peak how many reads align to that peak. And it will also calculate the RPKM or FPAKM values. And for those of you who don't know, that's reads per, sorry, I don't, reads per kilobase per million reads, I think. I'm probably wrong. Someone correct me if I'm wrong. And you will sometimes see FPKM instead, which is fragments, which is just from when you're doing paired end sequencing. You count a fragment rather than a single read, because a fragment is 2 reads. It's a bit pedantic, really, but you will see that. You saw that in the paper figure. They do FPKM, and it'll be the case with this tutorial as well, because we know they're double-ended. Unfortunately, when I was putting together this tutorial, I found some quite some significant performance issues with this step. That's actually not the fault of the Diffbind package. It's the fault of some of the bioconductor packages that it uses. Basically, the problem is that it will, if you have paired end data, it will read the whole BAM file into memory. This was causing me problems because it was crashing my workstation when I used the whole files. So if anyone is doing this analysis on a cluster or in a kind of big way, I'd be careful of this. Hopefully that'll be fixed fairly soon. And if anyone is interested in what those issues are, if anyone's like a bioconductor developer or something, I can point you to the GitHub issues if that's interesting. But I don't think that's like a generally interesting thing, because it's just very boring, really. But it's important, just to let you know. And that's partly why we use the downsampled files. When you get the tutorial and open the terminal, you'll see they're only kind of ½ gigabyte each of the BAM files, and that's just to get it to be able to run in a sensible amount of time. And this step will take a minute or 2 when you run it, so don't worry about that when it happens. Okay, so normalization. I'm not going to say much about this because it's passed down to DSeq2 by default, and if you want to learn about how DSeq2 normalizes data, it's quite a complicated thing, and there's good resources out there to learn that. Probably the DSeq2 vignette's a good place to start. But I've just given an example of an MA plot. It's called. It's kind of a kind of textbook RNA-seq analysis plot, just to give you some idea of how sort of complicated this normalization process can be. And if you look at the red line, it's basically saying that in different concentrations, there's different normalization factors. And that's what's really tricky about this. In some experiments, sort of stuff that's higher is amplified or suppressed, or stuff in the middle is too high or too low, and that kind of thing. So it is complicated, but we're lucky to have things like DSeq2 and EDSIAR to do this for us in a kind of systematic way, so we can get we can get robust results out. Again, this is all handled behind the scenes. And then finally, we basically run the model, or this has already happened by this point. I think it's run in one step with the normalization. But basically, at some point, you will have defined your model. So in this case, it's a very simple model, which is we care about tissue. And I haven't gone into detail in this because I think most of the experiments we'll be analyzing as part of AQUA-FAANG will be quite simple setups. But if you look at the vignette, they've got quite a complicated analysis where they have lots of different cancer samples, and you can use the contrast function to create sort of like more complicated models with different sets of linear effects, basically. Yeah, they go into detail about that in the vignette. But really, for you guys, that's handled automatically, and this is in the tutorial as well. But you can grab the significant peaks at the end using the DBA report function, which gives you a G ranges, which is a bit like a data frame of all of the significant peaks and all of the interesting things like the RPKM value and the fold change and the significance, the p-value rather. And I'll just put a note here. I think that's important because I think maybe in some of these analysis, if you've got a peak that's changed by a very small fold change, even if its cause is significant, that might not be that relevant for regulatory processes. We don't really know what it means to have a super high peak or a super low peak. It's not clear that that has a huge effect on regulation. But it's just something to think about if you're ever doing this analysis. So yeah, so that kind of concludes the run through what Diffbind is doing from like a kind of the point of view of the kind of the theoretically rather than kind of in the code. Has anyone got any questions about that?
Sorry, to me it was not really clear your last point with that the height, the maximum height or the minimum height of peaks doesn't matter.
I guess I mean it's not a straightforward interpretation in the same way that gene expression is.
So it's more of like if a peak is there, you may have to. So it's
you might want to. I guess what I meant was that you might want to try filtering, say, all peaks with a more than a 2-fold change or 3-fold change or something like that.
I see.
It doesn't really matter, to be honest. If you wanted to do a deep dive with the data, that might be a thing to do. But yeah, I mean, you will get a robust set of peaks no matter what with Diffbind, or you should do. So it's not really a problem.
Okay, thank you.
Okay, so obviously once you've got this list of differentially expressed peaks, you might want to say try and link these genes to enhancers, which is a really tricky thing to do. There's no two ways about it, and this is actually, I think, my role formally within AQUA-FAANG. We're looking at methods to do things like this better, to make interpretation of distal peaks better, which is not even really a problem that's been solved in kind of humans or mice, to be honest. So yeah, it'd be quite a general thing, and quite a general thing we don't understand. But there are some things you can do to make this easier. So I think someone's not on mute. But yeah, so one obvious thing would be to look at genes that are, when you have if you have an oocyte enhancer, you might want to look at not just the nearest gene, but the nearest gene that's expressed in oocytes, so any nearby expressed genes. That's probably important. Another way of looking at it is in genomic domains. So if you're lucky enough to have a species where you've got TADS or high C data, that's information about how the genome is compacted in space. That can be interesting because it's probably not the case that enhancers are contacting genes outside of their own TADS. There's also something called genomic regulatory blocks, which my old PhD group was involved in. And I think there's a couple of people here who work with ferrets, right? They may be familiar with this. I don't know. Sorry, Ferenc Muller in Birmingham, I meant. Yeah, so that's basically, there are regions of the genome where you have lots and lots of very highly conserved enhancers. And that seems to relate to TADS in quite an interesting way, basically. There's a paper from my group a few years ago, my old group rather, a few years ago that looks into that, which might also work quite well in AQUA-FAANG. I don't know, because you guys have some closely related fish species and that comparative analysis might be useful. So that could be something cool to do in the future. There's an interesting software tool called GREAT that was published a few years ago, which is basically looking at Go ontology enrichment for sets of enhancers within genomic domains and for nearby genes. And that's quite good because obviously assigning every single enhancer to its nearest gene isn't going to be right a lot of the time. But if you have enough of them, if you have a large range of enhancers, you might start to see some kind of Go ontology enrichment there that could tell you what they're doing. Although it might not be so informative. I'm sure if you did it with the oocyte enhancers, you'd find them related to, oocyte developmental processes, which would not really be that enlightening. But at the end of the day, if you have a variant that you're interested in, it's probably best to load up a genome browser and look what's expressed around the gene, look what's on, see if you have any kind of interaction data and that kind of stuff. It's still tricky. And also make sure you keep your mind open, right? It's not necessarily the case that this is going to go with the nearest gene or even a relatively close gene. I've just included a figure at the bottom for I mean, this is quite an opinionated idea. This is from a review a few years back. But the idea is that there's good evidence that there's plenty of enhancers that work like this. They'll be in the intron of a gene, but actually their target will be, hundreds of kilobases away in a different gene. And this is not at all uncommon. This seems to be kind of the norm. So it's just something to be wary of. I think most people know this. I think a lot of people do it anyway, just because it's the simplest way to get a paper out to say we linked the enhancers to the nearest gene and we got these results. But it's certainly possible to do better, I'd say. Right. So I think that concludes the sort of theoretical part of the discussion. Does anyone have any questions or just discussion points or ideas they want to share before we move on to kind of trying to get the code working?